Okay, we were talking about probabilities here, and there's actually a math component to all of these cross tests, and the outcomes are predictable statistically, and that basically is what one great piece that Mendel offered, and actually has been extended on with regard to the cross test and the breeding of plants and I guess you might say animals too. And one of the ways the statistics are calculated, particularly if you have a dihybrid cross, so you're looking at more than one phenotypical outcome. So you're looking at the alleles that are on separate chromosomes. You have this rule of multiplication, and so you have remember, you have the law of independent segregation and independent assortment. So those each are predictable in a 50-50 coin toss, that predictability. But what you do is you have this law, this rule of multiplication. So this rule of multiplication is the predictor that two or more independent events will occur together. And so the probability that two coins tossed at the same time will land heads up is 25%, is a quarter. So you have one coin toss 50-50 or 1/2, second coin toss 50-50 or 1/2. You multiply those by each other and you get the probability that the two being tossed at the same time will come up heads. And then if we extend this to genetics in terms of the rule of multiplication, probability that a heterozygous mix times a heterozygous mix will equal a homozygous recessive offspring is again 25%. So this statistically the chance for a particular offspring of a particular cross can be calculated. Then there's different types of dominance. So we talked about big R being the dominant allele and little r being the recessive allele, depending on what you want to use for that. But we find that there aren't always, that isn't the rules, doesn't always hold as a rule. Complete dominance can occur when phenotypes of heterozygote and dominant homozygote are identical. But you can also have incomplete dominance. And this happens at times where you have incomplete dominance. With incomplete dominance, what you have is something closer to the blending theory, but it still isn't blending because you're still going to have those separate alleles even though you have incomplete dominance. So with incomplete dominance, the phenotype of F1 hybrids is somewhere between the phenotypes of the two parent varieties. This is of the F1, but you still have two alleles, as I said, so it's not blending those. And then you have codominance, where you have two dominant alleles will affect the phenotype in separate distinguishable ways. And so the graphic that I have here makes it very simple, but with codominant you can have I guess a mixture or a variety of the two dominant alleles. One example of a dominant allele or a codominant allele, they don't tend to be, they're not necessarily more common. For example, looking at blood type, not only do you have a codominance with regard to blood types, but you also have the Rh factor. You probably know your blood type, or if you don't, it's a very interesting thing to find out. And maybe you're type B positive. That positive is the Rh factor, and that actually is a recessive gene. So dominant alleles aren't necessarily more common. They're just dominant and recessive. That's just the way we've labeled them. And with regard to blood, blood is considered codominant, and what you have is a protein marker on the outside of a red blood cell. That protein marker can actually be manifested as an A marker or a B marker, or you can have the A and B marker on a particular red blood cell, or you can have no marker on the red blood cell. So we have these genotypes where you can have a type A blood can have a genotype that's going to have an A dominant on both alleles, or an A dominant and then nothing. Or you can have B blood, which is the same, only with the B marker. You can have an AB blood where you have both the A and the B marker on the red blood cell, or you can have no marker, and that's where you get type O blood. So these alleles share the dominance, I guess you might say, if both of them are there, both type A and type B, or the A and B marker on the red blood cell. Other things that can happen with regard to genes, you can have a property that can affect multiple different areas of the body. We call them pleiotropy. And these alleles are responsible for affecting the organism, our organism. That's what we're speaking about here, and we're speaking about diseases such as cystic fibrosis and sickle cell disease. Cystic fibrosis is a disease where mucus builds up in the GI tract and also the lungs and makes it difficult to breathe, and also you have digestive issues. So this can actually affect unfortunately can lead to respiratory problems, can lead to digestive problems, but also with regard to sickle cell can lead to some blindness. The thing that happens with sickle cell is the red blood cell has there's a amino acid that has been changed and that change of just one amino acid causes the red blood cell to change its shape. Henceforth, you get the name of the disease, sickle cell. And this change in shape not only makes that red blood cell less likely to carry oxygen, it makes it more likely to clot. And so this clotting, unfortunately, this clotting factor can make an individual very, very sick, but also can lead to blindness, liver failure, and a heart attack. So some pretty severe effects of that particular allele. Another concept is epistasis, is the idea where you have a gene at one locus alters the phenotype expression of a gene on a second. And so we are all familiar with Labradors. We all love Labradors. They're adorable dogs. Well, you have your classic black lab, you have your chocolate lab, and then you have your blonde lab, or white lab, I guess it's called. And what you have is an allele that is responsible for color. The dominant one, B for black, and the recessive one, B for brown, but you have another allele that actually dictates whether there's any color at all. So you have to have both. If you want a black lab, you have to have a dominant B. You can have a recessive B also, but remember that's heterozygote dominant. But you also have to have the allele for color, so you have to have the capital C. So the two have to go together if you want that black lab. They have to go together. Recessive, you have to have a heterozygous, or the homozygous recessive, plus the capital C for color if you want a chocolate lab, and then no color if you have the lowercase c. It doesn't matter what the other ones are, you're still going to have no color. It's just not going to happen. There's no color that's going to be created with regard to that lab. Then there's also quantitative characters. These usually show up in polygenic inheritance. It's the additive effect, and this is the classic one that you look at with regard to skin color, where there's actually multiple alleles that dictate skin color, everything from the palest to the darkest. And what most individuals are a blend. And so if you look at these statistics at the very bottom here, the very palest the very palest skin colors, 1 in 64, the very darkest 1 in 64. But if you look at this as you go toward the middle, they're more likely to be a blend of the middle. So a blend of all of them. You have 20 over 64 right there in the middle. So these the quantitative variation, and usually in the additive effects when you have more than one allele that is dictating a phenotype. So that's a genetic allele dictating what you look like, your phenotype. And then the other thing with regard to a departure from Mendelian genetics can arise with environment. Mendel didn't take environment into effect at all in any of his studies, but he had enough on his plate, so there you go. But environment can affect the severity of some of the genetic inheritance that we have. For example, and a perfect example is these hydrangeas. Who hasn't seen hydrangeas? And whether you want pink ones or blue ones, what you're going to do is alter the pH of the soil that those hydrangeas are growing in. So you're actually shifting the phenotype, what that plant looks like, by altering its environment. And some of the inherited characteristics or the polygenic characteristics, some disorders are more highly affected by environmental factors than others. This didn't come from your textbook, but I kind of like this graphic where most of us have heard of muscular dystrophy. If one inherits the phenotype for the allele for muscular dystrophy, and that generally is a sex-linked, it usually is on males, then that is going to be expressed 100%. It doesn't matter at all on the environment. But if you go down here on the other side of the graph with a disease called scurvy, which we don't really have much experience with, but it's a lack of vitamin C, we think of that maybe as not even a genetic disorder. We think of it as so far from a genetic disorder that it actually is environmental. It's a matter of not having enough vitamin C at all. And so you have some variety with regard to how much a disorder is going to be expressed based on environmental factors. Humans aren't very good subjects for genetic research. We don't have, our generation time is too long, so it's very difficult to study from generation to generation. It's also very difficult to tell humans that they have to reproduce with specific people. There are laws against that. And culturally, experiments aren't very acceptable, appropriately so. But however, genetics are still looked at and used with regard to human diseases. What we do is we tend to look at past I guess, generations. I was going to say past breeding, but you know, we don't want to try to get too much into that kind of thing. But so past generations, and we do this in the capacity of a pedigree. We will look at your grandfather, your grandmother. I mean, when you go to the doctor's office, they will ask you, what did your mother die of, or what did your father die of, if they're still living. They'll ask you if they're still living first. And then if not, what did they die of? Brothers, sisters, if they had any particular disorders. This is looking at your family tree and making some assumptions based on your family tree as to what the probabilities are that you have inherited a predisposition for heart problems or a predisposition for Alzheimer's disease. We'll pick this up on the next one.